to introduce the last seminar of the year. I just remember also introduced the first seminar of the year. And those are both moments we crossed the threshold of 25 degrees where you get ice cream for everyone. So we start good, finish good. And with great pleasure, we got Professor Stefania Sardellitti from the University of Mercatore in Italy. And she's been working a lot in more abstract forms of signal processing that is interesting for people who work like topological data analysis. And she also liked lots of applications. She made a specific presentation for us relating topological signal processing applications into the brain. Thanks so much for coming in the forest.
Thank you, Fernando, for your kind introduction. And it is a pleasure for me to hold this seminar on the application of topological signal processing to brain networks. Well, why we need signal processing over topological space? We live in the era of data. Data are all around the house. Then in many fields, the main goal is to extract information from data for analysis, processing, learning, so on. Why it is a challenge to extract information from data? It is a challenge why data often reside over space that are not regular. The space are not able to be represented in a simple way. And these data, anyway, have a structure. Then our goal is to extract as much as possible the structure from the data. But to capture the relation among the data, we need also to represent data over spaces that are able to capture this relation. And we can employ the most general type of spaces that we have in this moment, and these spaces are topological spaces. What are topological spaces? Topological spaces are a set of elements with a set of neighbors relation among these elements. These neighbor relations are similarity relation between data that can be captured through a topology. The topology is the tool to encode the structure of the data to a neighbor relation among the data. There are several topological spaces: Euclidean space, metric space are topological space, manifold are topological space, particular topological space for which we have a local representation of the space that is an Euclidean space. We can approximate manifold locally to Euclidean space. In this figure, we have some example of a topological space. We have a manifold. We have a discrete topological space, simplicial complex, self-complex. This will be the object of our study. Discrete topological space. We analyze signal of these spaces. A challenge or the main goal is to extract topological invariant from data. Or our goal is to understand what are the properties of the space that are invariant to the formation of the space. Because it is important because we need to be able to understand when two topological spaces are equivalent. Okay, topological space are equivalent if some property as topological invariance are preserved. And example of invariance, connectness, compactness, fundamental groups. Fundamental groups are very important because they are able to capture the shape or the holes of the data. Well, when we have defined, when we define topological space to develop processing over this space, we need an algebraic representation of the space. Then we have algebraic topology. Algebraic topology provides tools to detect whether two topological spaces are homomorphic, are equivalent or not. For example, we have the Betti number. The Betti number counts the number of d-dimensional holes that we have in a topological space. For example, Betti number 0 is able to count the number of connected components of the space. Betti number 1, the number of holes, 2, the number of cavities. And for example, if we consider this particular invariant, the Betti number, we can observe that a point is different from the cube surface, because in the point we have one connect component, we have no holes, we have no cavity. But in the cube surface, we have one connect component, we have no holes, but we have one cavity. So that the cube surface is equivalent to the sphere surface because they have the same Betti number. We can say that these two topological spaces are equivalent. But the sphere surface is different from the torus surface because with the torus we have a different Betti number.
Why does the torus have two holes?
We have two holes, yes. We have two holes, this holes here around this red, this hole, and the other here.
That's not the cavity.
Yes, one cavity. We have two holes and two loops.
Two loops, yeah.
Yes,
Okay.
two loops.
Loops are one-dimensional.
Yes, yes. And one cavity because of the torus in this case is a torus surface, and we have a cavity. We have only the surface; it is not filled.
Okay.
In this example, of course. Okay.
Yeah.
There is some problem with the connection. Because I see connection loss.
Yeah, you are out. You have to-
Maybe it's problem of my PC, or it is a problem.
Are you on Ethereum network?
Okay,
What's
I think,
connection?
yeah.
And
Uh,
we can go over here and then you see the last deep swinger. That's it. Yeah.
mhm.
Please.
Not working.
because
You can.
but your works, maybe I can present it from your. You see?
Yeah, but it's not connecting in this computer.
Is there an internet cable?
Do you have your slides on PDF? You can present in my computer.
Yes.
Yeah.
Yes.
Yeah. I'm sorry. Like this.
Okay.
Here, yeah.
Okay.
Sorry.
Sorry for the convenience. Okay. Let us start from this slide. Well, the main concept is that we need topological invariance because we need to be able to distinguish topological space. We need to compare topological space so that if two topological space have the same invariance, these spaces are equivalent from a topological point of view. And from a topological point of view means that, for example, if we have a diagonal and we can deform it, we can obtain a cap. Of course, if we say cap, it seems different from diagonal. But from a topological point of view, these are the same because we can transform one in the other through a sequence of deformation that are also invertible. Okay. Another point is also to be able to find other topological invariants. Because if we have a high number of topological invariants, we are also able to enhance our representation of the space and are more able to detect if two spaces are equivalent or not. Okay. Topological data analysis. Topological data analysis is based on the analysis of data through topological tools. The goal is to discover the topology. Often we start from a set of point cloud, and these points are often in an Euclidean space. And the main idea is, for example, learn the topology by using the persistent homology. In particular, persistent homology is able to build ball around the poles, and then we increase the dimension, the radius of the ball, and we connect a point, and we connect triangles, and so on. But an important observation is that in topological data analysis, information comes from the property of the space, where no signal defined over this space. But an important question is, if we have a signal defined over this point, how we can exploit this signal to learn the topology? To answer to this question, we start from the most simple example of a topological space, the graph. In graph-based representation, we have data are associated with the vertices of the graph, and we have a link between two nodes if there is a pairwise relation between these two nodes. We are able to identify that there is a relation between the data observed over the two nodes. For example, in this case, we have a DNA nucleosome structure. Of course, in this case, there is a link if there is, for example, reaction, chemical reaction among the protein. If we have a graph, we need to define a signal process of a graph. We need an algebraic representation of the domain. We can exploit algebraic topology. We have the Laplacian matrix that can be represented as the difference between the degree matrix or the matrix with the elements on the diagonal are the degree of the nodes, minus A is the adjacencies matrix. We have an element that is different from 0 if there is a link from two nodes, and 0 otherwise. Another form to represent the Laplacian matrix is using the incidence matrix. The incidence matrix is able to capture the relation, the incidence relation between nodes and edge. And in this case, we can exploit this representation of the Laplacian matrix. We need to define an orientation. I don't go in the details of this representation. Well, we have our domain. We have an algebraic representation of the domain. We are able to define signal of this domain. Another signal is defined as the mapping from the vertices of the graph to a vector and a dimensional vector, real or complex. We associate with each node a signal. Then we have graph signal processing. Provide tools for the processing analysis of signal defined over the nodes of the graph. We have spectral theory, we have filtering, learning, and so on. But we are considering only the nodes of the graph, signal defined over the nodes of the graph.
Question. So you said that the nodes of the graph correspond to signals.
Yeah.
And so these signals, are they real value, complex value?
Yes, yes.
So they could be like RD vectors,
Yeah.
like d-dimensional vectors.
Yes.
And then the graph sort of represents some kind of relationship between these vectors.
The graph, yes, is able to capture the relation between the values observed over the nodes of the graph.
So if I were to think of these vectors as random vectors, then what does the graph represent? Does it represent like a correlation matrix? Or
In the case of a random vector, we need a proper tool to represent this relation, because we have a statistical dependence. We can also have graphical models that are able to capture the statistical dependence among nodes. In this case, if you have a graph, there is not a link between two nodes if these nodes are statistical independent given all the other nodes.
okay.
The partial conditional dependence. Okay. Okay. How we can use the algebraic representation of the domain? We need to use the algebraic representation of the domain to find the basis over which expand the signal defined over the graph. The representation is given by the Laplacian matrix, so that we can use the eigenvector of this matrix as a basis of the signal defined over the nodes of the graph. We can define a graph Fourier transform. What means a graph Fourier transform? Here, Fourier is an improper term, because we have Fourier transform only for graphs that are circular, for which the bases are exponential, complex exponential. In the other case, it is not Fourier transform. But it is the term that is used in the literature. We have graph Fourier transform. Is a simple representation of the signal as observed over the nodes of the graph over the basis given by the eigenvector of the Laplacian matrix. S hat are the coefficient according to which we combine the different vectors to obtain S. Here is important to remark that our goal is to find the representation of the signal that is sparse. What means sparse representation? Means that I wish to represent the signal with a small number of coefficients as hat. And in this case, if the graph signal admits sparse representation, the signal is called band limited over the graph. The bandwidth is the number of coefficients that I use for the representation. Now, I have a graph, I have a signal, I have found a representation of the signal over the graph by using a set of vectors that are orthonormal. And then I can use graph signal processing for filtering and so on. In particular, I only want to make this observation. If we have a graph composed by, for example, K cluster, the first K eigenvector of the Laplacian matrix are very smooth within the cluster. It is important because it means that if we are observing the signal over a cluster of a graph with a given number of clusters, we can represent this signal over the eigenvector of the Laplacian matrix. In this case, by using for example, only 3 coefficient, because we have 3 cluster. If I wish to represent this signal here, we can see that the signal is smooth within each cluster. I need only 3 coefficient to represent this signal.
So if I truncate the frequency basis by dropping, let's say,
Yes,
below
yes, yes, because we have in this
Then-
part of a course, we have the energy of the signal is concentrated over
So
this
how-
part.
how accurately can I then sort of reconstruct the graph? Is it gonna give me just the global structure like these 3 islands, or is it really also able to?
In this case,
capture?
we have, in this case, if I have this graph and I have with 3 cluster and I observe a signal that is smooth within this cluster,
Mhm.
the signal can be represented by using only 3 coefficient. The learning of the graph is another question.
Okay. But okay, let me rephrase the question. Is there a relation between sort of if I drop
No.
the small eigenvalues? Is there a relation in the error that I get in the signal with the amount of
This
eigenvalues
error is
that I
very
drop?
small, of course, because the signal is concentrated over this part.
Mhm.
Okay,
For example, if I plot just correctly first the lowest frequency eigenvector, so it will be only concentrated in one cluster, and so if I plot the second the next the subsequent graph frequency component, how can
the coefficient representation of the signal over these components is very small.
One more question. So when you say smooth, to me that implies differentiability of yours. What do you mean by smooth?
No, smoothness. Smoothness implies that if I consider the variation of the signal within the cluster, this variation is very small. In this case, if I take the difference of the signal of in the nodes within, for example, this this cluster here, the difference, the gradient, the discrete gradient within this cluster is very small because the signal are equal in this
I see.
case.
So
Okay, and the variation is between a cluster and another cluster.
that is why.
Okay. Indeed, I can show that we can minimize the total variation of the signal, but I don't go in these details for question of times. Okay. Well, why it is important to have a sparse representation? It is important because if I have a signal that is band limited, I can recover the signal from the observation of a small set of samples. It is important because, for example, we will see an application in brain networks where it is possible to learn the signal, the overall signal in the graph from the observation of the signal only over some electrodes of the networks. Okay, you can reduce the number of electrodes that you need to implant on the brain, because if the signal is band limited, you can recover the signal, the entire signal from the observation of a small set of samples. We can use this formula that this is a theory that has been developed in this work. Okay. The other step is that in many systems, brain, biological, social network, the complex interaction among data can not be reduced to simple dyadic pairwise relation because we have high order relation. Then we need tools to capture this relation. And what are the topological descriptors that are able to incorporate this higher order relation? We need to go beyond graph, for example, simplified complex, a complex, other graph, but in this talk I will focus on a simplified complex and set complex. And this example, for example, we see that the simplified complex is able to capture a triadic relation among the data by using the triangles, while a set complex is able to capture more sparse relation by using polygons. Well, the framework that we propose in this work in collaboration with Professor Sergio Barbarossa is a topological signal processing. Topological signal processing is a powerful framework combining signal processing and the topological tools for the analysis, for the processing, and so on of the signal. TSP generalized GSP to simplicial and self-complex. We can develop a spectral theory, sampling and recovering of signal defined over the complex, filtering, topology inference. Let us see in more detail simplicial complex. We can consider this a sample, photoshop networks. If I have a set of vertices, vertices are authors. There is a link if two authors have a joint paper. But to be able to represent if three authors are co-authored of a joint paper, we need triangles. We need to go beyond graph. Simplicial complex, this is the definition of the formal definition of simplicial complex. It is a finite collection of subset of various cardinality of the elements of a set of vertices, satisfying the inclusion property. We have this constraint on the topological space. The inclusion property, what means that if I have triangles, I have also all the simplicial of order of lower order the complex. Then we have edge and we have vertices. All couple of vertices, there is an inclusion. And these limits, yes, because in some application, I need more sparse representation. For example, I have a reaction among, here there is not the cell complex, but it is the same of the engram structure. Yes, it is empty in this case. It is this structure here, this cell complex here. And in this case, we can see that there are a sparse relation among the nodes, okay? If there is a polygon, this does not imply that there are all the edge between the vertices of the polygon. Okay. Why I don't understand why? It is There's a problem with this presentation. I don't know why. Why? Because it is all empty.
Huh. It's not supposed to be like that.
Now
A
it
few-
appears.
Few slides missing.
No, I
Hmm.
don't think why. Ah. There is a problem.
There was some graphic there. Supposed to be some
It
graphic.
is a, I don't know why.
Maybe because without computers, Linux sometimes doesn't show.
Oh, maybe. Maybe.
Maybe you can try to go back there.
Hmm?
It's, because I think that because of course we have maybe it is a problem with
Yeah.
Mac
that is-
that is not able to represent.
Mhm.
Um,
Did you
I can try
see
again.
if you have connection again? Internet? Yeah, because here there are more graphics. Uh-huh.
You enable my sharing
yes.
because
Good thing. Okay, now we should be able to go back. Let's hope.
Keeps you in the meeting, now.
okay. Okay. Then we needed to go beyond simplification complex. We needed to consider cell complex. Cell complex are more general topological structure that are able to capture a higher order relation. For example, we have the presence of triangles, of polygons of any dimension, and we call vertices 0 cell, edge 1 cell, and all polygons cell of second order. Simplicial complex are a special case of cell
So
complex.
these-
Yes.
You are drawing arrows, so they have a directedness. Also for cell?
In this case, the orientation is only used to define the incidence matrix of the complex. Okay, the simplicial complex is not oriented. Okay, the flow can go from V1, V2, and vice versa. Of course, there is an orientation. The orientation is only used to define the incidence matrix of the complex. Well, how can we define this incidence matrix? Of course, we have B1, the incidence nodes edge. We have B2, the incidence matrix between edge polygons. These metrics are able to capture the topology, the incidence relation. lower and upper incidence elevation between the cell. And then we have BT, the incidence edge triangles, edge square, and so on. In this case, we have an example of a cell complex. And another thing is that if we have two cells of a given order, for example, if I consider these two cells here that are one cell H, okay, C17, C16. These two cells are upper adjacent because are phases of this cell here, C12 of higher order. But they are also lower adjacents through this node here, C07. This is the topology that the matrix, the incidence matrix is decoding.
Lower adjacency means they share
Lower
variable that
Lower.
is smaller.
Yes,
Higher they are.
common phases of order lower, because the vertices is 0 cell, and the order is 0. I said I am considered the edge that are 1 cell. The order is 1.
Usually use tensors to connect levels that are not like one after another.
No, I am not using tensor. I have this matrix here.
So like,
This matrix here,
tensors.
B2.
yeah,
B2 is encoding is a matrix that
Yeah.
is able to encoding all the incidence between the edge and the second order cell. Second order cell can be triangles, square, polygons
Yeah.
of any order and so on.
I just expected that if we would go to bigger cells, like previously we also had matrices. So I expected that it would be tensors here, not just sets of matrices.
No.
And then probably equivalent.
No.
Maybe having the same work because the incidence like a boundary operation will be natural, and then everything is more natural this way.
I think that you can have a tensor if you consider upper graph. Okay. In this case, no, because we have a given number of edge. The number of edge are the number of row of B2. And each edge can be in upper, lower adjacents with the given cell. So that's why I needed to use a tensor. I can capture the topology
I don't say it is necessary to use them. It's just I expected them to see them here. Just me.
Okay. If I consider a cell complex, I expect a hierarchical structure. Okay, because I start from a set of nodes. From this set of nodes, I need to learn the pairwise relation that I can derive a graph L0. And I am in this case in the node domain because L0 is n by m matrix. We are in the node domain and we are able to process a signal in the node domain. We can go beyond it and we have L1. L1 is the first order Laplacian. And this Laplacian, in this Laplacian, we have a cell of second order. We are in the domain of the edge. So that I can represent this topology from an algebraic point of view using this operator, the first order Laplacian operator, L1. we have two parts. This part here, the first part is called the down Laplacian, because it is able to capture the incidence between the edge and the lower cell, the nodes. And the other part instead is the upper Laplacian, that is able instead to capture the incidence between edge and the upper cell, the triangles, square, and so on. It is important to capture the structure using a matrix. Of course, we can go on and consider a Laplacian of order k and so on. And another important thing is that we have an orthogonal condition between these two metrics. These two metrics belongs to two orthogonal space. We can exploit this property to represent the signal. Only to give you an insight on this new class of cell complex that we introduced in this paper, and we call this complex a planar hollow cell complex, because in this case we define in the planar case cell where that can also include holes. It is strange from a topological point of view, because from a topological point of view, we have that cell cannot contain holes. But anyway, we have proved that we can still extract the invariant property of the space by using this cell in the planar case. Well, we define a simplified complex, so we have defined the representation, algebraic representation. We can define signal of higher order of this space. We can define node signal, edge signal, and second order signal for a second order complex, of course. We stop to the second order complex. We can define a cell complex Fourier transform, a sparse representation of the higher order signal over the cell complex by using the eigenvector of the higher order Laplacian. And in this case, I can use this sparse representation for sampling and recovering of the signal, of the edge signal, or also of the cell signal. Depends on the complex that we consider. The representation of the signal of this space is the so-called Hodge representation of the signal. If I consider the space of the edge, I have the edge signal S1, and I am in the space of the edge signal Rn. And this space can be decomposed in three orthogonal subspaces. This decomposition take inspiration from the physics, from the vector field. And we have the representation of this partition of the space. We have the rotational flow. The rotational flow are associated to the image of V1 transpose. They are all the flow for which we can observe a divergence in the node. It is important because we can detect anomalies in the data, because there are some nodes for which we can have a source of information, sink, or source. Think if there is a loss of information or source of information. You have a divergence in the node. Or we have the other part that are solenoidal flow, for which we have circulation along the second order cell. Or the harmonic flow, for which we are able to detect holes. We have signal that are both solenoidal and the rotational signal. The divergence is equal to 0, the circulation is equal to 0, but these harmonic flow are able to identify holes in the topology.
These are the edge flows, like the signals in the edge.
Yes. And the dimension of the kernel is the number of holes in the cell complex. It is able to identify the invariant of the space, the number of holes in the cell complex, so that by using an algebraic tool, we are able to extract information on the space.
What does kernel mean exactly? Kernel of L1?
The kernel of L1, the dimension of the kernel of L1 is the number of holes in the complex. The cells that are not filled. What are the cells that are not filled? An edge flow then can be decomposed in a rotational part that comes to depend on the signal observed over the nodes. Indeed, it is the gradient of the signal, the discrete gradient of the signal observed over the node. This is a rotational part. It is a curve-free part with zero flow sum along any cell. If we consider the sum of the flow along the cell, it is equal to 0 for the rotational part. For the complementary part, the solenoidal part, instead, it depends on the cells that are present in the complex, and we can identify circulation of the flow of the information along the cell. And these are instead divergence-free, or means that all the flow in the node is equal to the flow outgoing from the node. And then we have the harmonic component that can be represented by using eigenvector in the kernel of the Laplacian. Each of these components can be represented by using eigenvector in the rotational space, eigenvector in the solenoidal space, or eigenvector in the harmonic space. How we can learn the topology of the cell complex? To learn the topology, we need to observe signal over the edge of the cell complex. We assume that the graph is given. If the graph is given, we need to learn only the second part of the first order Laplacian, B2 transpose. But the question is, we need to infer this second part. It depends on the data. Because if the data that we are analyzing have no component in this subspace, I don't need cell. I need cell only if the observed data, if we project this data on the orthogonal component to the rotational space or on the solenoidal subspace, this energy of the signal is different from 0, or is higher than a given threshold. Then the first step to learn the topology is to to check if we need to infer the presence of the cell. The second step is instead, if this energy is important, we need to learn the cell complex topology. Use this simple algorithm that is based on the minimization of the total variation. We minimize the circulation of the information along the cell. And we select the cell with the small circulation along the cell to infer the cell complex. This problem is not convex, but it admits a closed form solution. I don't go in mathematical details. Well, when we have find the topology, we can find also sparse representation of the observed edge flow using the eigenvector of the Fischer-Tropsch Laplacian. And to find this sparse representation, I observe signal y over the edge of the inferred cell complex. And then if I learned L1, I also have the eigenvector of the matrix. And I find the sparse representation of the signal, of the edge signal of this subspace. Okay. And I want a sparse representation, but I also have to impose a constraint on the goodness of the representation. I wanted to minimize this error, this data fitting error, or constrain this error to be lower than a given upper bound, given a small value. Using this band limited representation, I can recover the edge signal from the observation of the edge signal of a reduced number of edge. We propose this strategy in this paper here. Well, how we can explore TSP in brain networks? The challenge is that I have to capture a complex interaction and the structure that are useful to understand the functional activity or structure of the brain. There is a network neuroscience that is a subfield of neuroscience. Was aim is to provide the graphical representation of the interaction between brain regions or neurons. But again, in this case, we are using the pairwise relation. And topological data analysis is the tools that has been widely employed for the analysis of brain network. For example, using persistent homology, we can identify connected components, cycles, and cavities, and so on. But I think that TSP is a well-suitable tool for the analysis of brain networks, because in brain networks, we have a higher order interaction. Then group of brain regions or neurons are coupled through signals and work together to process information. That we have the processing of the information, we have the topology, and I think that this call for topological signal processing. We are observing node signal, we are observing edge signal, we are able to exploit all this information. And we are able to spot also the interaction that is dictated by the cell complex of the other order structure that we are using in the process. Well, the first application is on resting state functional connectivity signal. The method that we propose is learn first the brain functionality cell complex from edge signal, from edge data, in order to identify some brain region where there is circulation of the information. There are cycles. And use this learned cell complex topology to find the sparse representation of the signal and apply sampling theory to recover the signal from the observation of a small number of edge signals. This is the first application. We can see that I use this data set from UCLA multimodal connectivity database, and the edge signal is represented by the statistical dependence of the correlation between the signal observed over two years. And then we have, in this case, the structure of the graph. And the goal is to infer higher order relation or the presence of triangles, square, and so on. We need to learn the cell complex. The first step is project the observed edge signal on the solenoidal Placer-Modi subspace. Because if the signal has component in this subspace, then it is meaningful to learn the cell complex. Otherwise, I don't learn the simple complex. And we applied the minimum total variation algorithm to select the cell. In this case, we can see that there are a cluster of triangle signal, and there are also clusters of cell signal. In this particular data set, of course, I don't know why, but we can say that in the case of higher order cell, second order cell, we are able to capture circulation. Maybe there is an electrical coupling between this signal, and we need, of course, an interpretation of the result from neuroscientists. The involvement of neuroscientists is required because I need to give an interpretation to this result. What means that the circulation, there is a circulation information, means that we need to elaborate this information. We need the feedback between the nodes. I don't know.
You stop at squares, or you look at high order polygons?
No, in this case, I stopped to square for a question of visualization.
Do you use to define a signal the structural data or the functional data?
No, in this case, I have the signal observed of the edge that in this data set, the data set represents the correlation.
So it's functions correlation between the signal, the activity in each area.
Yes, observed over the nodes.
So, good clarifying question. So, how do you observe edge signals in this example? Like, I can understand that you place electrodes, then you're measuring node signals. But then how do you?
I don't know details on the on the processing of this signal, but in the data set, the edge signal are indicated as the correlation between the signal observed on the nodes.
But you do, because then if it's one individual, we have one correlation between a pair. You do it as signal for population. How do you get the signal?
I use this data set. I think that the correlation is derived by observing more signal over the nodes, I think.
Yeah. But so you only need the correlation between nodes to define the edge signal. That's what you're saying.
Because I think that a point in brain network is how can I observe edge signal? What is an edge signal in the brain network? We are able to observe a flow over edge. I don't know.
But also, when you talk about edge signals, there's almost like a temporality to the signal, right? Whereas with node signals, it's almost like you're thinking about a static.
Yes.
So how,
But we will see in the next example this part.
okay.
Okay. In this case, I have only used this data set to infer this self-complex topology and use this inferred topology for sampling and the recovering of the signal. For sampling and recovery of the signal. Then I find the sparse representation of the edge signal over the inferred self-complex. Okay, the sparsity versus the minimum square error is illustrated in these figures, where we have that with respect to a graph-based representation, we have an improvement in terms of trade-off between sparsity and data fitting error. So that if we add cell, we have more information, we are able to have a more sparse representation for given data fitting error. The use of cell provide an improvement of the performance.
So you say the more natural in terms of efficiency between the fit and the trade-off on the sparsity is the cell complex.
The trade-off is evaluated for the cell complex, the superficial complex, for graph-based representation.
And you say the best
Is for cell complex.
okay.
Okay.
What is the sparsity measuring? This is the measure.
Sparsity measure is this one. This. The number of the L1 or the number of coefficient that we need to represent the signal or the basis of the cell complex, superficial complex, or graph-based representation. It is useful to say that if you use a cell complex, we get more information with respect to graph-based representation with respect to superficial complex representation.
But if you added another dimension to the graph, namely computational complexity.
If
So you're looking at sparsity versus the mean squared error.
sparsity, yes.
But suppose that you added another dimension to this graph and you also asked, okay, what's the computational complexity of, computing these approximations? What would that look like? And I guess I'm asking, how hard is it to compute?
No, it is not hard to compute in the sense that
It's no harder than
that we
like
have
the
to
graph.
for example, if the complexity of the computation is only dependent from the fact that we have to identify cell. This is only the complexity of the situation, because on all this case, I am solving this optimization problem. I am minimizing the L1 norm with given bound on the data fitting error. We are minimizing sparsity with a constraint on the mean square error in all the cases. The complexity, I think that depends on only on the fact that we need to identify the possible cycles when we infer the cell complex topology.
So you're saying that is not a hard computation?
So the visual complex is usually hard. I don't know for the cell complex.
In simplification complex, we have only triangles.
Because you don't put more. You
I solve two triangles because it is a simplification complex. Simplification complex is not able to see square because we have only triangles. I am not able to feel a square. So that we needed to go beyond the simplification complex and we needed to use a cell complex. Because in cell complex, I can feel all polygons of any dimension, triangles, square, and so on. Okay. How can you use this sparse representation? I can use this sparse representation for sampling, so that I can recover the edge signal, the entire edge signal of the complex by the sortation of the edge signal only over some edge.
I understand it, but then for me, maybe I'm ignorant in this topic, that if I'm sampling, it's like if I would get a slice of the time series and then it should be similar to the full signal, but when you say you did your calculation only in one value of correlation, how the sampling can produce?
In this case, I don't see the time variation because I have the graph with the correlation of several of the nodes.
So your
In this case, I don't see the time. I don't see the time.
You're sampling a smaller number of correlations that.
Yes. Yeah.
Okay.
Okay.
I see it.
In this example only. Okay, you can see there is a good representation of the signal with the minimum error in the representation and the recovering of the entire signal. In this case, we have used about 70% of the edge, and we achieve a small normalized mean square error. Let us consider another application. We learn both the graph and the cell complex, in this case, the simplification complex topology, from electrocartography signal collected in an epilepsy study. We first learn the graph topology, and after the learning of the graph topology, we learn the simplification complex topology. The first, the learning of the graph topology, the sampling of this signal has been published in this paper to my co-authors, Professor Sergio Barbarossa and Di Lorenzo, while the learning of the simplification complex topology from a edge signal, we are working on this part, and I thank Lucia Tessa from Sapienza for her collaboration in this study. Well, we have this electrocartographic signal, and in this study we have electrodes that are implanted on the brain. We have 76 electrodes, and over each electrode we observed signal. We are observing 20 intervals before the onset of the seizure and 20 intervals after the onset of the season. In this case, of course, we are observing a space-time varying signal. But given the data matrix, we find a sparse representation. And we learn jointly the transform matrix U because I don't know the graph. So that I don't know the eigenvector over which I have to represent the signal. And I have to learn both U and S. But I learn the basis and the signal in order to make the representation of the signal over the learned graph a sparse representation. I know that the learned graph provides me a sparse representation of the signal. And in this algorithm, then we have to learn the transform matrix U, the sparse graph Fourier transform of the coefficient S, and the graph Laplace. In the first step, given the signal, the observed signal, we learn U and S, the eigenvector and the sparse representation of the signal. This is the first step. In the second step, instead we learn the Laplacian matrix from the sparsifying transform that I learned in the previous step by solving this convex problem. We minimize the total variation plus the two norms of the Laplacian with some constraint on the structure of the Laplacian. I don't go in the details of this algorithm, but we can find details on the paper.
What is this norm that you use? What's the norm have?
The Frobenius norm.
Means?
Yes, the Frobenius norm. Here we have the learned graph before and after the onset of the season. We used for all the graph the same equation to say there is or not a link. Okay? And we can observe here in this animation that before the season, the graph tends to be densely connected. When there is the seizure, the graph tends to lose connectivity, becomes more sparse. It seems that there is a loss of correlation between there is a disorder of the functionality of the brain. We can see this animation, and in the bractal phase it is strongly connected. Here it becomes less connected after the onset of the seizure. Here you can see again.
This is the connectivity over 1 second.
Yeah, yes,
Oh.
yes. Maybe there is a, okay, again, here in the bractal interval, we can see that it is strongly connected. Here it becomes disconnected. There is a loss of connectivity. Maybe there is a disorder in the formations. I don't know what happens.
But these are like the electrodes are distributed the same way these verses are plotted.
Yes, 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 yes. Yes.
So is there a interpretation? to
My
this?
interpretation is there is a lot of correlation. There is a noise, something that happens for which maybe functionality disorder
Oh.
in the activity, so that there is no link between the observed signal because there is no similarity between the signal observed over the nodes. The
Is that-
similarity is lost.
Is there a control theoretic interpretation? Like, it's almost like a loss of control in some sense.
Control?
Yeah.
Il control in what do you mean for
I
control?
mean, if it's densely connected, then presumably there's an exchange of information.
Yes, yeah.
Allowing you to somehow
Yeah.
distributedly control the firing of these neurons. But then you have this disconnection happening. So there's a lack of control. I mean, I don't know anything about it.
Yes, yeah,
Completely.
maybe, maybe.
Yeah, what is, oh, sorry, President, a mistake here. For epilepsy, before the onset, you have a hyperconnectivity, but hypersynchronization. But it's more known to the whole brain, and you have like just the electrodes, but it's very similar to what happens in the whole brain. Like before the
Interesting.
they call hypersynchronization, the combination of all signals get very strong before the signal. But I didn't, because it's very, one thing I think interesting was these electrodes are very noisy. It's not easy to make such a thing in the electrode level. Where I just left, she didn't tell more.
Okay,
Once she comes, you can ask her.
it can be an interpretation of this
She's not
disorder.
have any questions, so you can.
Well, is the inference meaningful to test if the inferred graph is meaningful? We apply sampling theory, and we recovered the signal observed of two nodes, in this case on the node 4572, and we can see that the recovered signal is very similar. There is a small error with respect to the true signal, so that the inferred graph is meaningful.
I have a question more like looking forward, like, because the on branch of network neuroscience, they make stochastic models for the brain. And the main goal is to the stochastic model have produced the signal, is training the real signal. So do you think that this would perform better than a stochastic model?
I don't know. I don't know if it performs better on the stochastic model. We needed to test this, of course, to say. But it is important that in this case we are able to reduce the number of electrodes to be in play on the brain, because we need only to observe over reduce a number of samples to recover the entire graph signal. And we go beyond the graph, we consider simplification complex. In this case, we need the edge signal. A common approach is a derive edge signal using statistical dependence, so the correlation between the signal observed over the nodes. I also find in this paper of Momami that electrical coupling among triplets of nodes associated to given triangles can be modeled by this triangle signal. So that I tried with this triangle signal. signal to see if we have a meaningful topology. And then we model the time varying edge signal by using the Hodge decomposition. And we use this last model to infer the simplicial complex topology. Okay. And in this figure, we have the learned simplicial complex before and after onset of the season. Of course, the graph is given. We have only to learn the simplicial complex by using the triangle signal according to the previous model. And the triangle color encodes the circulation along the triangle. We can see that when the graph is densely connected in the pre-ictal phase, the circulations are lower. When the graph is less connected in the ictal phase, the circulation tends to be higher and localized. We can see the animation. In the pre-ictal phase, the circulation is lower. In the ictal phase, the circulation tends to be higher. Why? I think that also in this case, we needed the interpretation of neuroscience. But it seems that when the graph is strongly connected, we have a low circulation of the information over the triangles. Because maybe, I don't know, maybe the graph is not, I don't know.
And it's one thing that is known, you should have come 1 week before because the expert in epilepsy was here. But it's known that where there is the source of the seizure, all the neighborhood around the source is damaged. So you can read more about that, how there is electrophysiology in the neighborhood of the damaged area. Because for a seizure, you have this the signal hypersynchronization, and in the tissue you have a normal tissue where there's the source of the seizure, like if they had to do inverse engineer to discover as the source, and the whole neighborhood is damaged. So it's an actual one homology that's put down.
Maybe.
And if you quantify exactly how is the homology around it.
Yes, maybe. We can see that here in the ictal phase, there are some triangles with high circulation in this region here. There are a small number of triangles, but they have a high circulation of information.
Yes, one other question, if you can identify with normal methods, what's the source of the seizure? Because then if this idea is correct, this area would be the more close to the onset of the seizure. Like that's more taken for the full signal, so you get your EEG before surgery until you get a seizure, and then you can get inverse calculation to which area of the brain is the start of the seizure. So if you have areas of high, strongly localized circulation, if this is true that these areas are really like covering the source of the seizure, it will maybe is targeting where is the seizure starting.
In this case, of course, I take the observation according to the study. I don't know where.
Yeah, maybe the publication they can tell in which, because you have several electrodes, if they use any method to then identify which electrodes the seizure started. Because then for the discussion, would be where there's higher circulation. Inside where the higher circulation.
Yes. The last result is on the node divergence. The node divergence encode if there are nodes that are sinks or source. It means that in some nodes there is a loss of information or is inject information in the nodes. And we can see that we have high divergence on some nodes. For example, I run the animation and we can see that there are some nodes in this region here in the pre-ictal phase for which the divergence is very high. It means that there is inject information, these nodes. And also in the ictal phase, we can see that in this node here, we have a high divergence. Maybe these nodes are source or sinks of information. Something is happening in this brain region. Okay. I think that topological is signal processing is the appropriate tool to capture the complex interaction in brain networks, so we can do sampling, filtering, and so on. We can learn the topology, structure feature. I think that we need to consider the spatial and time domain jointly in order to exploit, to capture the dynamics of the brain activity. Or also exploit this information and use a statistical framework. The involvement of neuroscientists is crucial for interpreting the result, of course, and for identify key features to be analyzed. I think that is a fundamental step. Okay. It's all. Thanks, and